Hello, everyone, and welcome to today's live broadcast, Single Cell Analysis of Virus Infection, Zika and Beyond, presented by Dr. Eric Clambey. We're excited to bring you this educational web seminar presented by LabRoots and sponsored by Affymetrix. Affymetrix eBioscience technologies enable multiplex and simultaneous analysis of biological systems at the cell, protein, and gene level, facilitating the rapid translation of benchtop research into clinical and routine use for human health and wellness. Affymetrix, part of Thermo Fisher Scientific, provides leadership and support, partnering with customers in pharmaceutical, diagnostic, and biotechnology companies and leading academic, government, and nonprofit research institutes in their quest to use biology for a better world. To learn more, please visit www.ebioscience.com. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would now like to introduce our speaker, Dr. Eric Clambey. Dr. Eric T. Clambey received his PhD degree from Washington University in St. Louis. He is currently an assistant professor at the University of Colorado School of Medicine, Department of Anesthesiology and Program in Immunology, and director of the University of Colorado Cancer Center Flow Cytometry Shared Resource. His research program focuses on the dynamic interface between immune system, infection, inflammation, and tissue repair. Please join me in welcoming Dr. Eric Clambey. I will now turn the presentation over to him. Dr. Clambey?
Good morning. Thank you, Christina, for that introduction. I'm Eric Clambey, and I'm at the University of Colorado, Anschutz Medical Campus. And today I'll discuss some of our ongoing research focused on single-cell analysis of RNA expression. My lab really focuses on the interaction between the immune system and infectious and non-infectious challenges. And today I'll discuss some of our ongoing work investigating viral gene expression in the context of diverse infections. So before I get started, I have no financial conflict of interest. It is worth noting that I was a recipient of the 2016 Affymetrix single-cell grant, and that mechanism gave me many of the supplies that we used for the talk I will discuss now. So today's talk is really focused around understanding RNA expression at the single-cell level. And while conventional assays typically to measure RNA typically focus on using pooled material where you're pooling tens to millions of different cells and then subjecting them to downstream analyses ranging from northern blot to PCR or microarray analysis. But a fundamental challenge with this method is that it provides an average signal of RNA expression. If you consider two different scenarios in which there might be comparable levels of RNA in that population. Here on the left, considering a population of cells in which there is homogeneous expression of your RNA of interest. Now, note that if you use a bulk analysis, you can't discriminate between whether there's homogeneous expression or heterogeneous expression indicated here. And for example, in this middle panel, where there is heterogeneity, you can see that some events are characterized by very low expression, whereas other events are characterized by very high expression. For example, this cell here in red. Now, this example focuses on a single RNA. But if you start thinking about a complex tissue where there are multiple cell types, or if you start thinking about multiple RNAs, you can quickly see that the complexity of RNA expression can really be obscured by using analyses in which you use pooled cellular material. Now today, we'll focus on some of our studies in which we've really focused on using a single cell method, flow cytometry, to analyze RNA expression. Now, importantly, these assays use gene-specific fluorescence to detect RNAs within intact cells. Importantly, by retaining the cellular context in which RNAs are expressed, this allows you to be able to define co-expression relationships with other RNAs, proteins, phenotypes, all at the single cell level. Something which is obscured by conventional pooled analyses. Okay. And so today we'll talk about two projects in which we've made use of this single cell methodology. First, we'll talk about our studies investigating Zika virus. In particular, which is a single-strand RNA virus, addressing the question, how uniform are viral RNAs at the single-cell level? In the second part of the talk, we'll focus then on a different class of viruses, the herpesviruses, to ask the question, how uniform is viral gene expression at the single-cell level? The goal in both of these studies is by analyzing RNA expression by flow cytometry at the single-cell level, we hope to gain new insights into virus and host RNA regulation during infection. So, much of today's studies will focus on use of this RNA detection platform, which we obtained from Affymetrix and eBioscience, particularly the PrimeFlow RNA assay. So this assay relies on branch DNA hybridization to achieve a high level of specificity and sensitivity, and is essentially a highly optimized fluorescent in situ hybridization assay to allow detection of RNAs at the single cell level. So in this assay, there are multiple steps. For example, with your target RNA, there are a series of multiple probes spanning your target RNA of interest, and typically RNAs are targeted by 20 to 40 probes that span your target RNA of interest. Building on this initial scaffold of the target RNA, there is then subsequent pre-amplification and amplification that allows for a pronounced amplification of the fluorescent signal. And so estimates are that there's approximately an 8,000 to 16,000 fold amplification of signal detection using this format. So the PrimeFlow RNA assay involves multiple steps. First, you have sample preparation, in which you can label your cells with antibodies, for example. You then fix and permeabilize these cells in suspension. Next, there is gene-specific. Next, you have gene-specific fluorescence, in which you incubate your cells with your target probes of interest. And importantly, this methodology allows you to detect up to 3 RNAs simultaneously in different fluorescent channels. Following this target hybridization, there is then signal amplification, followed ultimately by detection using conventional flow cytometry. And so if you think about what this system is affording you, this system allows you simultaneous detection of at least 3 different RNAs detected with different fluorophores. Now, the assays that we've used to date have focused on 3 fluorophores, Alexafluor 488, 647, and 750. It's worth noting that there is now a 4th channel available, which further expands the ability to combinatorially assess RNA expression. One really powerful thing about this is not only does it allow you to detect multiple RNAs, but you can also have simultaneous expression of protein expression at the single cell level. Another really beautiful thing about this method is it allows you to detect endogenous RNAs without RNA purification. So in total, this platform allows direct single-cell measurement of RNAs and it really bypasses two major limitations of conventional bulk analyses. First, it bypasses the need to pool cellular material, and secondly, it removes the requirement for standardization relative to a housekeeping gene. Okay. So the first part of the talk I'd like to focus on are our studies on Zika virus. Now, undoubtedly you've heard about Zika virus. There's a lot of concern in terms of Zika virus and its impact on public health. And Zika virus is an arthropod-borne virus, or an arbovirus. It is in the flavivirus family and is along with additional viruses including dengue, West Nile, and yellow fever viruses. Now, Zika virus is a positive sense single-strand RNA virus, and that's really one reason why we wanted to make use of this RNA analysis platform, the PrimeFlow format, because we wanted to be able to directly detect the viral genome, directly measuring the single-strand RNA in infected cells. Now, Zika virus is known to be transmitted by mosquitoes, but there's also now clear evidence of human-to-human transmission, including maternal to fetal transmission, sexual transmission, and blood transmission, which has raised many concerns. Okay. Finally, while Zika virus is associated with disease, and that occurs in approximately 20% of adult patients. So Zika virus-induced disease occurs when there are clinical symptoms in an infected individual, and that occurs in approximately 20% of adult patients. The symptoms for this include, in healthy adults, include fever, rash, arthralgia, myalgia, and conjunctivitis. And it's worth noting that there also are rare cases in which there are severe complications of Zika infection in adults, including individuals who develop Guillain-Barré syndrome. Perhaps the most notable and concerning feature of Zika virus-induced disease has been noticed in the context of birth defects. And now there's clear linkage between Zika virus infection and multiple birth defects, including growth restriction, intrauterine growth restriction, spontaneous abortion, and microcephaly. Now, one thing that's worth noting is that although Zika was identified in the late 1940s, it wasn't until the recent transmission of Zika to the New World that it's become a center of investigation, or it's become a topic of investigation. And so, while there's a lot of information about related flaviviruses, there's not as much information in terms of Zika virus itself. Now, Zika virus is like I mentioned, it's a positive single-strand RNA genome. Okay, and the genome is approximately 11,000 nucleotides long. Now, Zika virus encodes a single open reading frame, which encodes a single polyprotein, which undergoes proteolytic processing, and that's indicated by these to result in these multiple protein products indicated on the cartoon. It's also worth noting that Zika virus has both a 5' untranslated region as well as a 3' untranslated region. And these regions are highly structured RNAs, which promote both virus translation and replication. And so the question for today's talk that I'd like to share with you is how uniform or heterogeneous are Zika virus RNAs at the single cell level? Now, it's worth noting that we have focused on detecting the positive strand of the Zika RNA. We are now investigating studies to look at the negative strand, which is involved in replication. So for this first part of the talk, we focus on the detection of two different portions of the Zika virus genome. First, we have probes which can detect the Zika ORF, or open reading frame. Now, this is on the positive single-strand RNA genome, and the ORF spans a sequence of 10,216 nucleotides. This ORF encodes for the viral polyprotein, and it's also packaged as the virus genome, as is the entire positive single-strand RNA. The ORF is also a template for replication and translation. So that's the first target that we're going to focus on today. But we've also designed probes to measure the 3' untranslated region, or 3' UTR. Now, importantly, the 3' UTR is collinear with the ORF, meaning that they are expressed on the exact same strand, on the exact same RNA. And that's important to realize. The 3' UTR is a much shorter sequence. It's only 428 nucleotides long. But one reason that we're particularly interested in 3' UTR is it is a highly conserved element at the 3' end of Zika virus, and it has a high degree of secondary and tertiary structure. And so we were particularly interested in whether this highly structured 3' UTR might be differentially regulated in the context of cellular degradation of Zika virus genomes, and I'll talk about that in a few slides. Okay, so when we began these studies, we first made use of in vitro infection of vero cells, and vero cells are a highly permissive cell for Zika virus replication in vitro. We chose to infect cells using an intermediate multiplicity of infection, meaning that only part of the cells would be infected in the culture. And we did this on purpose so that we could discriminate between both infected and uninfected cells. And here what I'm showing you is flow cytometric analysis using the PrimeFlow format. And on the y-axis here, we're looking at side scatter or granularity. And on the x-axis, we are looking at fluorescence for the Zika virus open reading frame RNA, detecting with an Alexa Fluor 647 conjugated fluorophore probe. And what you can see is that in the left-hand plot, these are mock infected cells, and as you would predict, there are no cells which are positive for the Zika virus open reading frame, indicated by this paucity of events in the right plot. Now, when we infected cells with Zika virus, on the right-hand panel, we found that there was a clear positive population of cells which showed robust staining for the Zika virus ORF RNA probes, indicated by this red arrow there. So based on this analysis of a mixed culture of infected and uninfected cells, it's clear that using the PrimeFlow format, we can readily detect the single-strand positive sense RNA of Zika virus following in vitro infection. Okay. And we've also been able to obtain very comparable results when we've analyzed expression of the 3' UTR in Zika virus infected cells. So that's a single parameter, and that already affords unique insights because, for example, if you look at the spread of Zika virus ORF RNA, you can see that there is a spread in terms of how many of the relative intensity in ORF RNA positive cells. Now we wanted to increase the complexity, and here what we're doing is we're using the PrimeFlow system to simultaneously measure two Zika virus RNAs. So on the x-axis, again, we're measuring the Zika virus ORF RNA. And on the y-axis, we're measuring the Zika virus 3' untranslated region. As expected, mock infected samples on the left-hand side here have no positive signal for either the Zika virus ORF RNA or the 3' UTR. Conversely, when we looked at cells which were infected with Zika virus, again at this intermediate multiplicity of infection to give both infected and uninfected cells, we found a clear population of cells which were robustly positive for both Zika virus ORF RNA as well as for the 3' UTR. Now, one of the things that was unexpected from this analysis is we already know that the Zika virus ORF and 3' UTR are collinear, meaning that they're on the same positive strand of the Zika virus genome, and they're on the same RNA. And so one would predict that there would be a one-to-one correlation in terms of detection of the Zika virus ORF RNA and the Zika virus 3' UTR. One of the things that really surprised us was while we identified cells which had high expression of the Zika virus ORF and 3' UTR, indicated in this top right column, in the box highlighted in red, we also found a second population of cells which were positive for Zika virus RNAs, and those are indicated in this blue box. And these events were Zika virus ORF RNA positive, but had low to undetectable levels of the Zika virus 3' untranslated region. It's worth noting that in these mixed cultures, we also found a clear uninfected population which was negative for both Zika virus RNAs. And so one question that we're very interested in is what is the significance of Zika virus ORF RNA positive cells with a low 3' UTR abundance? Okay. So one of the beautiful things about flow cytometry is that you can analyze your different populations by multiple parameters. So here, for example, I've color-coded the 3 different populations that we have in these cultures, looking at infected cells in gray, the Zika virus ORF RNA 3' UTR low events in blue, and looking at the Zika virus ORF high 3' UTR high events in red. And when we look at those parameters, and we can then analyze them by additional fluorescent properties. If you look at the bottom, what we've done is we've done histogram overlays comparing the forward scatter or cell size and comparing cells with Zika virus RNA versus uninfected. And what you can see here, for example, is that there's a modest increase in terms of the cell size of cells which contain Zika virus RNAs. And that's true regardless of the relative expression level of the Zika virus RNAs. When we look at side scatter or granularity, we also find a modest increase in terms of granularity of Zika virus positive cells. Now, when you look at the Zika virus ORF RNA and Zika virus 3' UTR, what you can see is that there's really a wide range in terms of Zika virus ORF RNA and 3' UTR when you compare the red and the blue populations. And so one notable thing about this is that the blue population, which expresses the ORF but has low to undetectable 3' UTR, it has both decreased levels of Zika virus ORF RNA as well as the 3' UTR. And then finally in this analysis, we've included hybridization for a cellular transcript focused on actin RNA. And you can see that actin RNA expression is equivalent across all conditions, indicating that Zika virus infection has no consequence on actin RNA. Okay. So one of the things I mentioned was that Zika virus and flaviviruses are antagonized by certain cellular processes. And it's known that the flaviviruses have a highly structured 3' untranslated region. Now, importantly, studies by multiple groups have shown that there is a mechanism in place, a cellular mechanism in place, by which a cellular exonuclease, here referred to as XRN1, is a protein which is capable of degrading flavivirus genomes. And what happens when XRN1 degrades Zika virus genomes from the 5' and 3' end is that XRN1 is capable of degrading through most of the flavivirus genome. However, importantly, when it reaches this highly structured portion of the 3' untranslated region, this region is so structured that XRN1 is incapable of completely digesting this highly structured RNA. And as such, the 3' UTR of Zika virus and other flaviviruses contain XRN1 resistant elements. And the reason that that's important is that during flavivirus infection, what happens is that you have an accumulation of subgenomic fragments of flavivirus genome which are not degraded by XRN1. And here you can see the accumulation of some of these 3' untranslated region fragments which resist XRN1 degradation. So with this in mind, we wanted to ask the question, what is the consequence of depleting XRN1 on the frequency and magnitude of cells that express the Zika virus RNAs? And we'll look at both the ORF RNA as well as the 3' untranslated RNA. And so when we've done this, we've made use of, again, in vitro infection. Here we're using A549 cells in which we've made use of RNA interference. And we have two conditions. We have, on the left-hand side, we have cells which were transfected with a scrambled small interfering RNA. On the right-hand side, we have cells in which we've depleted cells of XRN1 using an XRN1 siRNA. In this assay, we have two conditions that we're comparing. We have mock infection, and we have Zika virus infection. So here, for example, in the second plot, we're looking at cells which have been transfected with a scrambled siRNA and infected with Zika virus. And what you can see is that 33.8% of cells in this culture are positive for Zika virus ORF RNA, as detected by PrimeFlow. Now, it's also worth noting that there's clearly a spread in terms of the range of intensity for the ORF RNA positive cells, with some cells being low and some cells being high. Interestingly, and to our surprise, when we depleted cells of XRN1 using RNA interference, we found that there was only a modest change in terms of the frequency of cells which were Zika virus ORF RNA positive, going up to 39.1%. But then look at the intensity of these ORF RNA positive events. They're really focused in terms of a low expression of the ORF RNA. And conversely, there's essentially a dropout of cells that were Zika virus ORF RNA high. Now, you can quantify that by flow cytometry, and what you can see is that there's approximately a twofold or 50% reduction in the mean fluorescent intensity of Zika virus ORF RNA when you consider cells which are Zika virus ORF RNA positive. Okay? So this result was really surprising because the paradigm in the field is that XRN1 degrades the Zika virus genome. And so if anything, we would have anticipated that by depleting XRN1, we would have had an increase in the amount of Zika virus genomes that we could detect. But that's clearly not what we're seeing here. So two major take-home points from this analysis. One, Zika virus infection is associated with a range of intensities for the ORF RNA. And XRN1 depletion has a modest effect on the frequency of infected cells, but it decreases the overall abundance of ORF RNA within infected cells. So we're really interested in these observations. and are actively pursuing two major questions here. One, why are there varying intensities of Zika virus ORF RNA in infected cells? Is this an issue of kinetics or genome amplification? And what's the relationship to the 3' untranslated region? We also really want to know what happens when you deplete XRN1. Why is there reduced genome amplification? It's worth noting too that XRN1 regulates both cellular and viral processes, and so it's possible that the effects that we're seeing here are secondary due to some other perturbation that occurs following XRN1 depletion. So what I've shown you thus far is focused on the Zika virus ORF RNA. So let's turn to the 3' untranslated region. So one of the things that was very surprising with this was here we're focused on analysis of the Zika virus 3' untranslated region. Again, measuring it by the PrimeFlow. And so on the x-axis here, we're measuring the Zika virus 3' UTR. Again, on the left-hand side, we're looking at scrambled siRNA transfected cells versus the right-hand side, in which we're focusing on cells which are XRN1 depleted. This analysis revealed another surprise about the interaction between Zika virus and XRN1. So if you look on the left-hand side, you can see that 14.6% of cells are positive for the Zika virus 3' UTR following transfection of cells with the scrambled siRNA. Okay, so those are our control cells. But look at what happens when you deplete XRN1, and that's over here on the right. And what you can see is that we significantly increase the frequency of cells which are positive for the 3' UTR. And so we're going from 14.6% of cells which are 3' UTR positive in the scrambled condition to 29.3% in cells transfected with XRN1 depleting reagents. Now, notably, the mean fluorescent intensity is quite comparable between these two conditions. And so, based on this, our conclusion is that XRN1 depletion results in an increased frequency of cells with Zika virus 3' UTR. And there's really minimal changes in terms of the overall abundance. And so, we're getting more cells which are positive for the Zika virus 3' UTR following XRN1 depletion. Okay, so that immediately raises multiple questions. First, well, what we'd like to focus on next is, why is XRN1 knockdown associated with more Zika virus 3' UTR positive cells? One possibility is that there are altered RNA species generated during XRN1 depletion. We're also very interested in whether XRN1 degrades the Zika virus 3' UTR in some infected cells, but not in other cells. Okay, so kind of broad strokes of what we've seen in the context of Zika virus. So first, we have the observation that there appears to be a discordance between the Zika virus ORF RNA and 3' UTR. And that's depicted here again on this flow cytometry plot. And in this analysis, we've identified that there are cells with high expression of the Zika virus ORF and 3' UTR. But we've also identified a population which is characterized by lower expression of the ORF RNA and low to negative expression of the Zika virus 3' UTR. So how do we make sense of this? Well, our working model is that we think that these two populations reflect different stages of virus infection or replication. So, for example, in the context of these double positive events, we suspect that there is a molar excess of the 3' untranslated region relative to the Zika virus ORF. In contrast, we think that Zika virus ORF RNA positive cells, which are 3' UTR low, likely have a greatly reduced frequency of the 3' UTR relative to the ORF RNA. And this is something that we are actively investigating now. Okay. Based on this, we would posit that the double positive events really represent cells which are late in virus replication, possibly with large accumulation of the 3' UTR or the or the sfRNA, whereas the ORF positive 3' UTR low events might be either early in the virus replication cycle, or alternatively, it could be that there is 3' UTR sequestration in replication complexes. So future goals for this include really hammering out the kinetics of these different populations. For example, is there a stepwise progression from these low events to these double positive events? We also are very interested in gaining molecular insights into what RNA species are present and that we're measuring. And ultimately, we'd like to know what RNAs are present in the single versus double positive events. So in terms of the role of XRN1 and Zika virus infection, our observation is that XRN1 depletion gives us two unanticipated changes. One, it reduces the abundance of ORF RNA within infected cells. And two, it results in an increased frequency of 3' UTR positive cells. Now, our working model at a broad level is that XRN1 modifies Zika virus RNAs to promote Zika virus replication. So what do we think is happening here? Well, we think that in the context of an XRN1 sufficient cell, indicated here on the left-hand side, that XRN1 is capable of degrading many Zika virus genomes, as well as misshapen or irregular 3' UTRs, indicated here in blue. Ultimately, however, XRN1 is incapable of degrading many of the 3' UTRs, and that's why you have this accumulation of 3' UTRs indicated here. Now, conversely, in the context of XRN1 knockdown, we think that there is likely altered processing of the Zika virus genome, and whether that results in a reduced genome amplification or virus replication. We also suspect that there might be aberrant RNA species that are generated following XRN1 depletion, and whether that's a direct consequence of XRN1 depletion or a secondary consequence of XRN1 depletion remains to be determined. Okay. So thus far I focus on our analysis of Zika virus, which is a plus-strand, single-strand RNA virus. So next I'd like to focus on a short analysis of herpesvirus infection. And this is a very different type of infection, but I think our studies illustrate some of the real power of looking at RNA analysis at the single-cell level. So herpes viruses are completely different from Zika virus. They're large, double-strand DNA viruses that can result in multiple outcomes. And so herpes virus infection can result in two widely divergent outcomes. One, it can result in something called lytic infection. And during lytic infection, there is abundant viral gene expression. There is new virus produced. But today what I'd like to emphasize and show you some data on is a process referred to as virus-induced host mRNA degradation, or host shutoff. These viruses are also capable of establishing something referred to as a latent infection, and that's indicated here on the right-hand side. And latent infection is characterized by really limited RNA expression. And so with these divergent outcomes, we're really interested in how RNA is being regulated at the single-cell level. Now, let's talk about this virus-induced host shutoff. So what is it? Well, during lytic replication by many herpes viruses, there is degradation of both viral and cellular mRNAs. And importantly, this virus-induced host shutoff involves both viral and host nucleases. And so if you look here, the first step in this degradation process occurs through a viral endonuclease. But importantly, following initial cleavage by viral endonucleases, cellular exonucleases come in and participate in degradation of these target RNAs. And you'll note that XRN1 is one of these exonucleases involved in degradation of these RNAs following during lytic replication. Okay, so we wanted to study this process of host shutoff using a murine gamma herpes virus 68, and this is a mouse animal model for the human gamma herpes viruses, including Epstein-Barr virus and Kaposi sarcoma associated herpes virus. One strength of the gamma HV68 system for the purpose of this talk is that this virus is capable of establishing a very robust lytic replication system in vitro, which is characterized by this virus-induced host shutoff. And below is just a schematic of the genomic complexity of gamma HV68, the virus that we're studying. Okay. So, like I mentioned, herpes virus infection is characterized by divergent outcomes, whether a lytic infection or a latent infection. And importantly, latent infection, while it's transcriptionally quiescent, is capable of being induced to induce reactivation and to generate new virus progeny. So we wanted to make use of the PrimeFlow platform to analyze these processes. And so what I'll show you now is some of our analysis in which we focused on 3 targets. So first, we have looked at First, we've looked at RNAs, focused on viral short non-coding RNAs. And these RNAs are required for optimal gamma HV68 pathogenesis in vivo. And importantly, they're abundantly expressed during virus replication. We've also analyzed beta-actin both as a cellular messenger RNA, but also it is known to be a target of the viral endonuclease, and so it is known to be degraded during virus infection. So the top 2 targets are RNAs. We've also focused analysis on a viral protein, which is a regulator of complement activation, and we're using that as another marker of the progression of virus replication. So here what we're going to make use of is in vitro infection systems to study the dynamics of RNA and protein during herpes virus infection. And so the first thing I'd like to show you is where we have performed de novo infection of murine fibroblasts with gamma HV68. And this is a process that results in lytic replication. Now, we've done this and analyzed actin expression through multiple parameters. In the left hand, or in panel A, here we're looking at relative expression of actin by a conventional assay. Here we're using quantitative real-time PCR, and what we're comparing are cells which are mock infected, which should have normal levels pardon me, of actin, versus cells which are infected with wild-type virus, or cells infected with a virus which is deficient in the viral non-coding RNAs. Okay, and this is the rightmost is simply a control virus for these studies. So this is conventional analysis, and what you'll notice is you see approximately a 50% reduction in the amount of actin standardized relative to 18S, which is an RNA which is impervious to host shutoff. So that's a conventional assay, but now let's look at another type of analysis, which is this PrimeFlow format, and this is in panel B. And so here what we're doing is we're using probes to recognize the actin mRNA, the cellular actin RNA, and what we've analyzed, what we've compared here is cells in which there's no probe, and that's the top, versus mock infected, which is the second line, versus wild-type gamma HV68 infection, or this viral non-coding RNA mutant, the TEAM or TKO on the bottom. And what I'd like you to notice here is that whereas mock infection gives you a uniform positive peak for actin, when you look at cells which are virally infected, not only is there this high population of actin, but there's a second population which is characterized, which is actin low, And so the beautiful thing about this is that rather than obscuring this variation in terms of actin expression by pooling cells which are both infected and uninfected, analysis of actin at the single cell level using PrimeFlow really allows us to discern that there's a bimodal distribution of actin, and that's illustrated in panel B. Okay. So that's a single parameter. So let's ratchet the complexity up a little bit more here. And if you look in panel C, this is looking at a two-dimensional analysis in which we've focused on actin expression on the y-axis versus these viral small non-coding RNAs on the x-axis. And what you can see here is that many of the cells present in these cultures are characterized by high levels of actin expression. These cells are also low for the viral small non-coding RNAs. Now, conversely, if you look at cells which are have expressed high levels of the viral small non-coding RNAs, these events also are characterized by very low expression of the actin RNA. And there's a third population here, which is characterized by intermediate expression of the viral small non-coding RNAs, but also has normal levels of the actin RNA. And so just by looking at two parameters, the expression of two RNAs and their relative expression to one another, it's able to reveal that there are multiple populations of cells which you would have never been able to identify using conventional analysis. Okay. And so this is an example where measurement of actin mRNA levels can reveal this virus-induced host shutoff. And importantly, this process occurs only in a subset of virally infected cells. And these are the cells which are characterized by high expression of the viral small non-coding RNAs. We've also made use of detecting these viral small non-coding RNAs to identify rare populations of cells. So here what we've made use of is a cell line in which there is uniform expression. The prediction is that there will be uniform gene expression. And what I'd like to focus your attention on, this is a B cell line which contains gamma H368, this virus. And what we've done here is we've used PrimeFlow to measure the viral small non-coding RNAs. And in the uninduced state, this cell line has about 20% of the cells which express the viral small non-coding RNAs. Now, when you stimulate these cells with chemical stimulus, this chemical stimulus induces reactivation. And here what we're focused on is these cells treated with TPA, which is a pharmacologic inducer of reactivation. And when you do that, you'll notice a couple things. First, the overall frequency of cells which express this intermediate level of the viral small non-coding RNAs is fairly similar between the untreated and treated cells. The thing that really pops out as a difference, however, is that there's a new population of cells that are characterized by high expression of the viral non-coding RNA. And so by using this technique, we're really able to focus on a rare population of cells which are characterized by a unique expression signature of high viral non-coding RNAs. What's really cool about this is, and exciting, when you compare the properties of these cells based on differential expression of the viral small non-coding RNAs, if we compare cells which are either virus non-coding RNA mid or bright, what you can see is that cells that express high levels of the viral non-coding RNAs, which is in this open black line, are characterized by a prominent population of cells with low levels of actin. Okay? And so by using this single cell analysis, we're able to focus on a population in which 1% of the cells have a phenotype, and then we can say, what is the actin profile in the rare population of cells? Okay. And so using this method, we're able to show that the cells that express high levels of these viral non-coding RNAs have multiple characteristics of lytic replication. They have decreased actin RNA, and they have increased viral protein expression. So from our studies on the herpes viruses, we've been able to show that virus-induced host shutoff can be measured at the single cell level, and it occurs in only a fraction of virus RNA positive cells. And so we're actively investigating why there is kind of a spectrum of RNAs, of viral RNAs, and with only a subset of cells experiencing this actin low phenotype. We also have made use of this system to investigate an inefficient process, that being reactivation from latency. And if you think about what we've shown here, we basically have really focused on a rare population of cells, and by comparing the uninduced versus the induced condition, we've been able to identify this lone event here, which is characterized by a really unique cluster of phenotypes. So if you think about the big picture of what I've talked about today, we really focus on single-cell analysis of viral and host RNAs by flow cytometry. We've been making use of this methodology to study both Zika virus, which is an RNA virus, and for those studies we've been able to directly measure the Zika virus genome in infected cells. And we've also been able to begin to reveal unexpected complexities in terms of the regulation of these RNAs, particularly in regard to how they're potentially degraded by host exonucleases. We've also made use of this method to interrogate rare populations of cells, for example, in the context of looking at reactivation from latency, in which there's only a rare frequency of cells which have a phenotype of interest. But and this study really wouldn't have been possible by conventional analysis using pooled cells. It would have really obscured any changes that we'd seen. By analyzing 3 parameters of 3 different RNAs, as well as cell size, granularity, and a single protein, we've also been able, in the herpes virus system, to identify a whole series of different phenotypes in which cells are characterized by a spectrum of RNA expression. And so the real take-home from this is that single-cell analysis of virus and host RNAs really affords new opportunities to understand biology. We think that this methodology allows us to understand variability in response to therapies and to potentially identify rare subpopulations of cells that might be either uniquely susceptible or uniquely resistant to treatments of interest. And ultimately our goal in this is to understand the extent and the factors which drive variability in RNA expression at the single cell level. Ultimately, to be able to intervene and identify unique therapeutics to target both rare virally infected cells, but also you can easily imagine turning this to other types of analysis, including the study of cancer, for example. So finally, I'd like to acknowledge people who facilitated this study, including Andrew Bessler in my lab. Importantly, these projects are really a close collaboration with 2 labs. Dave Beckham, who's an investigator who studies Zika virus, and he's a practicing infectious disease doctor. And so all of the studies with Zika have been in intimate collaboration with the Beckham lab, without whom we couldn't have done these studies. For the herpes virus studies, that's been a close collaboration with Dr. Linda Van Dyke and Lauren Oko in her lab, who have greatly facilitated the studies. And I should say that Aaron Massey in the Beckham lab has been critical in allowing the PrimeFlow analysis of Zika virus infection. We have funding from the American Heart Association, NIAID, and the Department of Anesthesiology. And finally, I'd like to thank people at Affymetrix and eBioscience who have been great in terms of giving new insights in terms of ways in which we can really optimally use the PrimeFlow format to get new biologic insights. Thank you very much.
Thank you, Dr. Eric Clambey, for that informative presentation. It's time for Q&A. So if you have any questions you would like to ask Dr. Clambey, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click the send button. We will answer as many of your questions that we have time for. Okay, let's get started. Dr. Clambey, our first question is, what types of targets are detectable by PrimeFlow? Are there any limitations on what can be detected?
Sure. So the question is, what types of targets can be detected by PrimeFlow, and are there kind of special considerations or things which might not be readily detectable? So we've made use of PrimeFlow to detect a variety of targets. And it's worth noting that this platform really allows you to not only look at mRNAs, but also non-coding RNAs. So thus far, we've been able to detect multiple cellular mRNAs of varying length, and the smallest mRNA that we've detected has been probably about 500 base pairs long. When we've analyzed non-coding RNAs, we've been able to, the Zika virus 3' UTR is 428 nucleotides long, and the viral small non-coding RNAs, there are multiple of these RNAs, and so we're detecting a cocktail using a cocktail of probes. But it's worth noting that each of these viral non-coding RNAs is just about 100 base pair nucleotides long. And so the long and the short of it is that this system is very capable of detecting shorter RNAs. We have not tried detecting microRNAs. There is a published reference to this from Parikis et al., published in Nature Communications 2016. Also, the new, my understanding is the new formulation of the PrimeFlow that will be released soon has also been optimized to potentially facilitate detection of microRNAs. It's, one thing I would like to point out is that the RNAs that we're detecting are fairly abundant, especially the small ones. And so that might be one consideration for some of the studies. If you have a very scarce or low detected RNA, that might be pushing the limits of detection. That being said, Parikis et al. Again have shown that this system is capable of detecting down to 10 copies of RNA per cell. In terms of structures of RNAs, the processing of the material is fairly stringent, and so at least many cellular secondary structures are going to be degraded or melted by these hybridization conditions. One question we do have that we're investigating now is whether a highly structured viral RNA, such as the 3' UTR, might have some regions of inaccessibility, and we're doing that using kind of not different probe sets to interrogate binding capability of different probes.
Dr. Clambey, can you combine the PrimeFlow analysis with downstream analysis, such as cell sorting?
Yes, thank you for the question. We're very interested in integrating the PrimeFlow platform with downstream analyses. We have not yet done that. But if you think about essentially the material that you're left with at the end of this, you can run it through a conventional flow cytometer and readily detect those events. And so, for example, the ability to sort cells based on differential RNA expression, certainly I can't think of any reason why that would not be technically possible, and that's something that we're actively developing. One thing to note is that you won't be able to purify viable cells. And so if, for example, you wanted to sort viable cells based on differential RNA expression, currently the PrimeFlow platform does not allow that. Nonetheless, I think that there could be a lot of informative studies in which you could sort different populations of cells based on differential RNA expression, and then, for example, subject them to quantitative PCR or to Western blot or to RNA-seq. And we've been actively discussing this with Affymetrix, and we're now investigating the possibility of treating PrimeFlow treated material essentially like formalin-fixed paraffin-embedded samples in which, with the appropriate conditions, you can actually extract RNA. And so kind of the next wave of studies that we're doing will be analyzing the ability to sort cells and then to purify RNA using kits which are optimized to recover from very stringent preparation conditions such as formalin-fixed paraffin-embedded samples. But I think it certainly should be doable, and Dan Kaufman's group just published an analysis in which they sorted cells and they quantified HIV proviral DNA following PrimeFlow. So I think it certainly is something that will come online and really allows even more exciting opportunities.
Thank you, Dr. Clambey. We have so many great questions coming in, but we are almost out of time. This will have to be our last question. Dr. Clambey, what is the impact of secondary, tertiary RNA structure on probe hybridization?
Thank you for the question. Yes, so certainly one thing to note about the PrimeFlow system is that it's really designed to achieve high target specificity and sensitivity by using a series of probes that span your RNA of interest. The reason that I mention that is if you think about an RNA that where portions of that RNA might be highly structured, you could imagine that at least some of the probes would efficiently hybridize to unstructured elements in those RNAs. Now, in terms of structured RNAs, such as the Zika virus 3' UTR, clearly, while that is known to be a highly structured RNA element, clearly our probes, and there are only 5 probes that target the Zika virus 3' UTR in our probe set, clearly they are able to detect, to sufficiently hybridize and detect Zika virus 3' UTR. Now, one thing that we are actively investigating is whether, in the context of XRN1 depletion, where we see an increase in the frequency of cells which are Zika virus 3' UTR positive, one possibility is that perhaps some of the increase in frequency that we're seeing are due to kind of misfolded 3' UTRs. So one way that we're hoping to address that is basically by combining some of the cell sorting approaches that I discussed earlier, and then to clone those RNAs or sequence those RNAs to identify whether there might be point mutations in the 3' UTR. But the bottom line is that while structure in extreme circumstances might antagonize probe hybridization to some degree, clearly it is not a major issue given our robust ability to detect both the Zika virus 3' UTR. I would also like to point out that the virus small non-coding RNAs that we've detected in the context of herpes viruses, those RNAs are also highly structured, and they include a hybrid between a tRNA-like molecule and microRNA stem loops. And so, especially in the context of small non-coding RNAs, I think that the system has been very robust for us in allowing us to gain new insights that would have been very difficult to do by conventional bulk analyses. Thank you for the question. I appreciate it.
I would like to once again thank Dr. Eric Clambey for his presentation. Dr. Clambey, do you have any final comments for our audience?
First, I'd just like to say thank you for your attention. I really appreciate it. We've had great success with this analysis platform, and I think I'd like to just emphasize that some of the insights that we're gaining really wouldn't be possible without by conventional assays. And so certainly while this approach doesn't answer every every question. I think it's really a unique tool in a toolbox, in the scientist's toolbox, that can really provide new insights that you wouldn't be able to obtain by other methods. So I'm happy to take additional questions in the future. And again, thanks so much for your attention.
Thank you once again, Dr. Eric Clambey. And thank you to our sponsor, Affymetrix, for making today's broadcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May 10th, 2017. You will receive an email from LabRoots letting you know when this webcast is available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.